All right. Well, hi everyone. My name is Varshini Balaji. I am a intern at NASA Ames, part of the 2026 BMSIS YSP project, and today I will be presenting on what I worked on this summer, which was focused on designing an astromycology kit for undergraduate and high school students. So just a quick background. What is a science kit and why do we need them? Science kits are prepackaged materials, equipment, and supplies that are used to conduct simple experiments, often aimed towards high school and undergraduate students. Their purpose is to be used as a learning tool in school as they give students a more hands-on approach to the science that they may be learning, almost supplementing the lecture content that they are already learning from. And research has shown that utilizing science kits in school curriculum actually leads to a greater retention of information, as the more hands-on learning of scientific concepts causes them to stick more in the brain, according to Foley 2013. And as you can see on the right, there's a picture of an example chemistry science kit. And the purpose of my project is to design a science kit specifically for astromycology and metrology. Some quick definitions. Astromycology is the study of mycology and its relation to light in space, while metrology is the study of measurements. So again, the purpose of my project is to design a kit that teaches and exposes students to the basic academic concepts of these fields and hopefully get them more interested in these fields of research. So again, the purpose of the astromycology kit is that it is aimed towards a wide range of students and hopes to get them more exposed to the field and more interested in the field. So the main concept of our kit was that it would be aimed towards a good range of students. It's aimed towards both high school and undergraduate students. And the way we accomplished this was by having the same experiments have different levels of complexity. For example, high school students would have a more introductory science level with more simple experiments, which would be focused more on teaching these core astrobiology concepts such as, wow, life can survive beyond Earth, and also teaching them the importance of establishing a metric system for measuring results. For undergraduate students and graduate students who could also use these kits, the experiments will be more focused on developing an experiment in the astromycology field and applying the concepts they've already learned into these fields. They would also focus their metrology studies more on the repeated calibration of experimental tools and also statistical analysis of uncertainty in measurements. So by adding these different tiers, one kit can be used towards many different student levels and is almost multi-purpose. So I proposed two general experiments that could be put in these kits. Obviously, more can be added with time, but these were two experiments that I think kind of encapsulated the main ideas of astromycology and metrology. The first experiment was a stress tolerance experiment, where fungal strains would be grown in different stressful conditions, such as UV exposure, saline exposure, pH range, heat ranges, etc. And students would also go ahead and design their own measurement system to test or to test how growth would be measured. So they could look at the radii of strains, they could look at how long they've grown, how thick they grew. That is up to the students. And this teaches them the basic metrology skill of how do you go ahead and measure your results. And the deliverable results that they would have to give to their instructor would be a simple growth chart of 7 to 14 days to show how the different strains would grow in these different conditions, and a short written report on the difference in growth between a control strain and a more extremophilic strain, or a strain that likes more extreme environments, and which of their measurement methods work best. They would also have a feature where they could upload their results to a common database along with other schools so they could compare their results with other schools and study the difference in growth in different environments. The second experiment is more focused on a project the lab is already working on, which is mycotexture growth. For some background information, mycotexture or mycology architecture is the use of the mycelium of mushrooms to create architectural structures. So on the right here, you can see an example of a cube that was formed with mycotexture. So in a mold, mycelium along with soil we pack in, and the mycelium will grow in that shape. So the second experiment would focus on growing these small mycelium cubes in a provided substrate and mold, and would be used to build simple structures to test stability. So mycotexture in the lab I'm working in actually has a lot of application in architecture for space. Buildings, there's a potential of building bases on the moon using mycotecture due to their resistance to many space conditions. So students would use this experiment to study mycotecture and mycelium's resistance to conditions such as UV radiation and lower temperatures and compression. So they would go ahead and grow these structures up and then use them to build test structures such as example spaces that could be used to then test their resilience against space-like environmental stressors, showing them the alternatives to traditional architecture. And once again, their deliverable would be like a mock project report that they would turn in talking about the growth process, the observations they saw, and then factors such as mass loss and shrinkage before and after the stressors, as well as what other variables may need to be tested. So right here is a basic equipment list that I compiled that would be needed to be included in these experiments to conduct these tests. There's obviously some individual equipment such as a UV dosimeter, which would measure UV radiation, which I'll get to, and mycelium mix, but there's also PPE, worksheets, regular markers. These kits often need a lot of equipment. And now I want to touch a bit on the methods that I used. Obviously, it's one thing to design kits, but we have to also test to make sure the experiments we design are actually functional and deliver the results we want them to deliver. So for my stress experiment, two strains, a control strain and a more extremophilic strain, were grown on PDA, potato dextrose agar plates, that were then altered to expose these strains to various stressors. So for my control, we chose Saccharomyces cerevisiae or baker's yeast, while for the extremophilic strain, we chose Rhodosporidium toroloides. S. cerevisiae is a strain that has been very documented. There its ranges in survivability. Its pH ranges, its temperature ranges, its salinity ranges are all well documented in literature, which makes it a great control strain since we already know what it can and cannot grow in. Rhodosporidium toroloides is a more thermophilic strain that has some documented tolerance to pH and salinity ranges. So through this experiment, we hope to not only look at or look at in more detail the ranges it could survive in, but also study its use in these experiments. So since it's already an extremophile, it would be a good example to show students an example of a strain that can survive in not so or some more unique conditions. And I want to emphasize here that unfortunately I was only able to come into the lab in person for 3 weeks. So I was working on a very limited amount of time. So I wanted to make sure that I was prioritizing the experiments that I thought would be most important for the kit. So the four main stressors I chose to handle in these 3 weeks were UV exposure, salinity, pH, and heat. And you can see the ranges right here. And for my mycotecture experiment, we focused mainly on trying to grow up the cubes to see the difficulty of growing the cubes for someone who doesn't have any experience in that, which would be me, and then testing their strength in a typical lunar day-night cycle. So Ganoderma lucidium, which is the mushroom or mycelium that is grown by the lab of mycotecture, was grown up and then placed into cube molds to form a cube shape, and then exposed to 6 hours of 120 degrees Celsius, 6 hours of room temp, and 6 hours of -80 degrees Celsius to replicate the lunar weather cycle, just to test its resistance and test an experiment that students could do with these cubes. So now I'm going to go a couple slides into my results. So first, I want to draw your attention to these plates right here, which look at the resilience of Saccharomyces cerevisiae and Rhodosporidium toroloides at pH of 3, 5, and 8, respectively. Unfortunately, we did find out that my R. toroloides colonies were all contaminated. Almost every single tube except the first one was contaminated. So most of my R. toroloides results were not accurate. So right here, you can see there's a great amount of bacterial contamination in the rightmost plates, but we were able to study the Saccharomyces tolerance, which is still a good benchmark. So as you can see here, this pH of 3, Saccharomyces only grew at the 10 to the 0 and up to -2 dilutions. Uses 10 to the 0, -1, -2, 3, 4, 5. Versus at 5, it was able to grow all the way up to the 10 to -4 dilution. At pH of 8, it was able to grow up to 10 to -4 as well. This shows and aligns with the literature results of that S. cerevisiae has a pretty wide pH range, but leans more towards the basic side of a 5 to 7 to 8 pH as a good survivability, versus pH of 3 kind of falling into a pH range that it cannot survive in. And This also shows pretty crystal clear results that outside of these pH ranges, SC or S cerevisiae is not able to survive, as right here, even though there is some growth at a pH of 3, it's not as much as at the pH of 5 and 8. These next set of plates look at the tolerance of salinity. Once again, the Rhodosporidium toroloides were contaminated. There is some growth in the 10 to the 0, but as these were the same colonies that were grown on the pH plates, I would like to err towards the idea that this is unfortunately bacterial contamination. And meanwhile, the Saccharomyces cerevisiae plates showed a pretty interesting drop off as salinity increased, with 0 to 4 and 6% having good growth. 6% starts to show a drop off at 10 to -1, and 10% barely has any growth at 10 to the 0. This shows that an increase in salinity leads to a decrease in survivability for Saccharomyces cerevisiae, once again establishing it is a good control. The next slides here look at Saccharomyces cerevisiae and Rhodosporidium toroloides at different temperature levels. These were actually the only Rhodosporidium toroloides colonies that were able to grow without contamination. So these results are accurate. And as you can see here, the Rhodosporidium toroloides is able to grow at all 3 temperatures, including 40 degrees Celsius. And meanwhile, the Saccharomyces cerevisiae is able to grow at 30 and 37, but is not able to grow at the 40. This shows that Saccharomyces cerevisiae A is once again a good control, as it shows a drop-off in more extreme temperatures, but B, the Rhodosporidium toroloides is a great thermophilic strain that can be used to show that some strains have more tolerance for high heat and may be able to survive in more hotter temperature planets, such as Mercury, something that students could kind of connect and think about. So there's even an interesting morphological change here, as the strains do get darker as it gets more hot, which could have some indication that students could think about the melanin and what that may contribute to heat survival as well. And finally, these are the UV assay tests for up here, Saccharomyces and Rhodosporidium down here. Unfortunately, once again, these were the contaminated strains, so these results are all bacterial contamination. And just to draw your attention, the UV exposure times were at an intensity of 2.65 watts per meter squared at 254 nanometers, something that I measured, and students would go ahead and practice their metrology by using a dosimeter that will be provided in the kit to measure the intensity of their UV light that they use. This would be, again, very important in establishing accurate results and measurements as well. And Saccharomyces cerevisiae is control. This first half is 5, second half is 10, 20, and 30. And as you can see, there is a drop off as the UV exposure increases. 5 and 10 is around the same, or 10 minutes around the same growth patterns. However, at 20 minutes, there is less growth, and by 30 minutes, there is quite less growth, even though these all have the same amount of colonies plated. This shows that Saccharomyces cerevisiae does have less UV resistance and drops off over time. Next, I want to go over my results for the mycotexture experiment. So this picture right here shows the Ganoderma lucidium cubes after 1 week of growth in the candy mold. And then these cubes were then taken and baked in the oven at 120 degrees Celsius for 6 hours. This is how they looked after that. And then they were put in the -8 degrees Celsius freezer for 6 more hours, which is how they looked right here. And in between each stage, they were weighed to measure difference in mass. We did not have the equipment to do any strength testing, so we mainly looked at the changes in morphology as well as in mass. And right here is a quick table I put together with the data of the mass changes. Half these cubes were frozen and then baked first, and half were baked and then frozen first. So in this case, the data I want to put up here was the cubes that were frozen and then baked first. So as you can see here, the cube mass at first, average weight's around 1.95 grams, pretty normal. After freezing for 6 hours, the average weight drops down to 1.3 grams with an average weight loss of 0.65 grams. Of course, these are averages. Each number is going to be different. I just want to find some concise numbers here. And then after they were baked, the average weight drops down even more to around 0.55 grams with an average weight loss of 0.75 grams. This shows that as they undergo the conditions, there's some shrinking and weight mass loss as well. We could not do any strength testing, but I just arbitrarily crushed and put some force on the cubes, and they were able to hold their structure as well, which indicates that there may potentially be a resistance to stressors even resistance to stressors, and structural integrity even after the intense temperature changes. And then finally, I want to go a bit into detail about the fungal mycelium preservation experiment I conducted. So as mentioned in the previous experiment, the mycotexture experiment, or previous slide, mycotexture experiment was very successful because we were able to grow these cubes up. As a complete beginner, I was able to grow and conduct these tests, showing that they're easy enough for high school and undergraduate students to conduct, and they would be able to learn these core concepts of mycotexture and their importance to space travel. However, in order to conduct these experiments, it's really important that clean, uncontaminated cultures of G. lucidium are transported to students. If they are not transported, other strains grow in the spawn bags as well, and the molds, then they may lose structural integrity and the data they collect may not be accurate, which would ruin the experiment. So in order to make sure clean cultures can be transported, also tested a method from a paper by Montserrat in 2019, which used a method to preserve mycelium from Humphreyia coffeata, and which is a medicinal mushroom. And G. lucidum is also a medicinal mushroom, so I want to test if this method would work. And the method involved growing a plug of G. lucidium on PDA plates that had sterile filter papers or cellophane papers, which you can see here, placed on the media before. So the mycelium would grow over these papers. So when after a week of growth, you remove these papers and place them in sterile DI water, the mycelium would be stuck and grown in the papers. That way, when you place the papers on fresh new plates, there would be fresh new cultures that could grow from this mycelium. And when I conducted these tests, and the original test was conducted over 120 days, I did not have that time, so I just conducted it over 3 weeks instead to see the growth after preservation. And the results, which I will show in the next slide, showed that preservation actually was possible, but only when you have extremely sterile water. I had used old DI water that was sterilized, which led to a lot of bacterial contamination, as you can see here. However, mycelium was still growing inside the papers and preserved. I did not have time to plate these new myceliums, or I had not time to plate these new papers like entirely. These are only a couple of the papers I plated. However, there was contamination, but with contamination also came a growth of mycelium. This shows that it is possible to preserve through this method Ganoderma lucidium, but we need to make sure the DI water is extremely sterile to prevent any other bacterial mats forming. So, my conclusions, why does this matter? First of all, my experiments showed that Saccharomyces cerevisiae is a good control strain for stress assay tests. It has a normal pH range survival, it has a normal salinity survival range, normal temperature, and UV exposure survival range. It does not go out in the extremes, so it serves as a good benchmark for students to compare extreme tolerant strains to. It also has a very strong growth pattern. There was no trouble growing it, no contamination, no issues. This experiment also showed that Rhodosporidium toroloides has good potential to be an experimental strain. Even though there's a lot of contamination, the heat assays showed that it does have a greater resistance to heat and can be used to compare against Cerevisiae to show a strain that has a more extreme or tolerant tendency. Our experiments also show that mycotexture is an easy to grow and test experiment that students can conduct. As once again, I was a total beginner. I did not know anything about mycotexture, but I was able to conduct this experiment, and this is something that students would be able to conduct as well. And also showed that G. lucidium can be grown and preserved in this DI water method as long as the water is sterile. So overall, even though there was only a couple weeks of wet lab work, I was able to draw a lot of important conclusions for this kit. The next steps for this experiment, first of all, obviously, is to test stress assays with non-contaminated R. toroloides strains. Though we know through literature the conditions they will survive in, we want to make sure that the experiments we're conducting give the results we want students to see. So we need to conduct these tests with R. toroloides that don't have contamination. Next would be to design a full protocol for all the experiments. This is something I've been working on, but until we have those R. toroloides results, we cannot nail down a full protocol. And then once that protocol is designed, we'd go ahead and greenlight the design of a full kit, including the materials necessary and acceptable for use by both high school and undergraduate students. We don't want them spending a lot of money beyond the kit, and we don't want them using equipment that would need them to have a lot of different training. The bottom line of this kit is that it's an accessible and cheap way for students to learn about astromycology. Thank you so much for listening to my presentation. I had a great time during this internship. I would like to take some time to thank people who've helped me with this internship work. First of all, I'd like to thank Dr. Lynn Rothschild Lab for hosting me during the YSP program. They gave me a lot of access to lab equipment, time, mentorship. Even if I was running an experiment, I had no idea how to do it. They were always there to help me and give me a listening ear. I would like to especially thank Dr. Rolando Perez, who was my mentor for the summer, provided me with a lot of guidance and a lot of help throughout this project. I would also like to thank the BMSIS YSP program for giving me this opportunity to intern at NASA Ames. This is something that I've only dreamed about, and to have do this summer was an amazing opportunity, and even complete my own independent project, something I never would have dreamed about. I would also like to give special thanks to Graham Lau for assisting me in joining the YSP cohort this year, as well as providing me with a lot of support, including deadlines, extensions when I would have a hard time with this video. So thank you so much to everyone who's made this project work, and thank you for listening.